So thank you so much, Jeannie, for that kind introduction. As Jeannie said, my name is Joe, and I'm a graduate student in Rockefeller University. So here's just a brief outline of what I'm going to do today. I'm going to take you through what I'm talking about and why I think it's important, and then give you some background, which is important for understanding the paper today by talking about immune memory, before we dive into the paper itself and talk about what the authors did in this particular paper, and what they showed, and what questions I think remain outstanding in this kind of field. But before I do that, a little bit more about who I am. So this is a picture of Joe the scientist. As I was introduced, I'm a third-year graduate student at Rockefeller University. I work in a field called biophysics, which you could really think of as the interface between biology and physics, the physics governing the biological molecules in your body. And I moved to the United States 6 years ago from London, where I grew up, and I was born. Cool. I'm not only a scientist, as was alluded to. This is a picture of me from this Christmas, actually, where I was hiking a mountain, which is one of the things that I really like doing: skiing, backpacking, being in the outdoors. Okay, but enough about me. Let's talk about what I'm going to talk to you about today. So what am I talking about, and why? So this is the paper that I'm presenting to you today. It's called Lack of Infection in Rhesus Macaques Infected with SARS-CoV-2. And as was discussed prior to me starting, this is a preprint, which means it's not been certified by peer review. So I'm just going to take you through a little bit more about what that means and elaborate on what Pedro and Jeannie were saying at the beginning. Basically, how science is done, if you start out on the left here, you can see that scientists collect scientific data, obviously, and we write it up in a report. And that report usually is evaluated by other scientists in this process called peer review, where they give us feedback, tell us experiments, maybe extra experiments that we should do, caveats we should include, etc., etc. And there's a feedback loop there. And then the end product after that of that peer-reviewed report is hopefully published and hopefully has an impact, a positive impact on society. This process of peer review, though, can take some time. In fact, in some cases, it can take up to a year or more. And that's one of the things that's currently a bit of a problem in science. And especially in the context of a pandemic, where we want policymakers and the general public to have up-to-date information as quickly as possible, this process of publishing material immediately online is really useful. And that's what a preprint is. For those of you who have been following the styles of scientific reasoning in this series of talks, today I'm going to be talking about an experimental evaluation where we're using data to test hypotheses and get to an answer as a result of that. So why am I talking about this? Well, the first reason, of course, is that there's considerable interest for people who have had COVID-19 because they want to understand their own personal risk going forward. Is it possible for me to get sick again? And that will set to rest many fears that people have, or cause people to behave differently, possibly. But more importantly, it's got profound public health ramifications. And that's because depending on how regularly people can be reinfected by a virus, different control measures and strategies are going to be appropriate. And that's because, you know, if people are susceptible to being infected again shortly after being infected, it means we're going to have to think differently about vaccination. It means we're going to have to think differently about social distancing and other public health measures. One final thing before we start talking about the background to this paper is that what I'm talking about today is research done on monkeys, and that deserves acknowledgement. Whenever scientists work with animals, we think about three big things, what we call the 3 R's. The principles to reduce the number of animals used in our experiments, refine our experiments to maximize the well-being of the animals in the experiments, and wherever we can, replace animals with other systems, be they computational systems or other systems like cells in a petri dish. And also to replace more sophisticated animals with less sophisticated animals. We'd rather do experiments on flies than on monkeys. But in some cases, as with this paper, it's important that we do this work, and we need to understand it in as close a relative to humans as possible. There are lots of organizations involved in thinking about these ethical concerns and monitoring how we use these organisms. And I've just listed some of them here. They are the university itself, government regulators, and professional organizations which certify them. And this paper comes from a lab which is certified by this organization. That's the gold standard in scientific animal care. Okay, so before we talk about the paper, we need to talk a little bit about how immune memory works. And this is not really news to any of you, because all of you will know from your own experience that your immune system remembers past infections and stops you getting sick for a second time. What I think of when I think of this is how relatively unconcerned my parents were when I got chickenpox, right? Because adults know who had chickenpox when they were kids that they're really unlikely to get it again from their children. Their immune system has remembered the virus that causes chickenpox and makes them very unlikely, inoculates and protects them against getting sick a second time. It's also obviously the basis of vaccination. When we vaccinate people, what we're doing is we're introducing a modified, weakened strain of a virus or a pathogen, or we are introducing a fragment of that pathogen to cause the immune system to remember it, so that when you see it in the flesh for real, your immune system prevents you getting sick because it's remembered the pathogen or remembered the sort of mock version of the pathogen. So what's going on under the hood here? How is your immune system remembering things? Well, it's helpful to break the immune system down into the two branches of it. So we have what's called innate immunity in humans, and we have also what's called adaptive immunity. And innate immunity is a general rapid response, which even starts before you are infected with a pathogen. It's all the defenses like your skin and mucus in your respiratory tract that stop you being exposed to pathogens in the first place, as well as general defenses when you get hit by a pathogen. Adaptive immunity is slower, and it's specific, and it involves the production of specialized antibodies and specialized immune cells. And I'm going to talk more about what those immune cells and antibodies are. But this is the branch of the immune system which is able to remember things. And so when we talk about immune memory, we're talking about the adaptive immune system, and we're talking about a very specific memory of a very particular pathogen. So how does it work? Well, what happens is a pathogen such as a virus infects us. We have a process going on inside the immune system where we learn the identity of that pathogen, and then we produce specialized molecules known as antibodies. They're proteins, and we produce specialized cells which make those proteins and do other things to help us become immune. And those molecules and those proteins are present and help us clear the infection, and they also are present going forward to help us protect against the same infection in the future. These are the three key players. We've got antibodies, we have B cells which make antibodies, and T cells which help B cells to make antibodies and do a whole load of other things. And obviously many a PhD thesis has been written about any one of these things, so I can't really take you through them in detail, but I just want to give you an overall outline of kind of what's going on. So antibodies work by being sticky, and I've highlighted the bits of the antibody which are sticky. I've just circled them in white here. And really you can think about them working in two ways. They work in lots of ways, but two really important ways they work is by sticking to a virus or a pathogen inside your body and sort of waving to the rest of the immune system, over here, I've stuck the bugger, you know, you can come and get him. And they also work by sort of gumming things up, right? If you're a virus and you get stuck all over by an antibody, you can imagine it's not really very easy for you to move around, for you to do what you want to do, to interact with the players you need to interact with inside the body to cause an infection. And B cells are obviously going to make the antibodies, and T cells are going to support them in that role. And they are also very specific. The bits I've circled on the T cell and the B cell, like the antibody, are sticky, and they're sticky only in a specific way. So an antibody that's sticky against the measles virus will not be sticky against a different virus like flu, right? And that's really important. It's a specific response. So what happens when you remember something? Well, we can think of it a little bit like this. So along the bottom here, I'm just plotting advancing time. And initially what happens in my example here is you get infected on the left. There's a blue arrow. And your immune system takes some time to respond to that, your adaptive immune system. You could think of it as it's finding the right antibody. It's calibrating a response. It's figuring out the best way to clear this virus, this pathogen, away. And so there's this time that elapses, and during that time, you're feeling unwell, right? Because the pathogen is running wild, is infecting you. And that's what being sick is, right? So then you make your antibodies, you make your B cells, you make your T cells, and those help you clear the infection away. And then subsequently, if you get reinfected with the virus, your immune system reuses the same cells and the same antibodies it made the first time. There's no point reinventing the wheel, right? It's already done this before. It knows how to get rid of this particular pathogen, the correct antibodies, the correct B cells, the correct T cells to use. And that, as a result, it's able to deploy them very quickly to help clear the infection away. But your immune system is actually even cleverer than that, because it doesn't just use the same things again, it uses the same things again in a more strong and extensive way. So if we plot, again here I'm plotting time along the bottom here, and I'm plotting, going up the side, the strength of response. After the first infection, you have a weaker response that occurs after a larger period of time. Whereas after the second exposure, you have a stronger response that occurs after a shorter period of time. So your immune system is reusing the cells and the molecules it generated the first time, and it's deploying them harder and faster to help you beat the pathogen more quickly and prevent you going through that period of getting sick. Okay, so can I bring this to life? How can I make this a bit clearer to you guys? Well, I have an example or something of an illustration. So if you'll indulge me for a moment, I have a problem for you. And the problem is this. Maria went to the bread store to buy a loaf of bread for dinner. She had 2 quarters, 4 dimes, 3 nickels, and 2 pennies, and the total cost of the bread was 82 cents. She promised to make sure she had exactly 1 coin remaining after the purchase. So the question I'm setting for you is, which coin did she have left after buying the loaf of bread? And I think Jeannie is going to set that up. So you should be able to answer that question now. I'll give you a few moments. The votes are trickling in. We've got about 20 of you so far. I'll give you a little bit longer.
We forgot to warn people to bring their scratch paper to the talk.
People are getting there. People are getting there.
Yeah.
Okay, cool. I think we'll leave it there. I've given you a minute. So what we can see is you did get the answer right overall. It is a quarter. That's the coin she had left over. Great. I'm really glad you did. Now I have another problem for you. Another question for you. Maria went to the bread store to buy a loaf of bread for dinner. She had 2 quarters, 4 dimes, 3 nickels, and 2 pennies, and the total cost of the bread was 82 cents. She promised to make sure she had exactly 1 coin remaining after the purchase. Which coin does she have left after the purchase? Now, I'm not actually going to ask you to do this, because hopefully you've noticed that it's the same question, right? So hopefully if I was to ask you to do this, you would just remember that you'd solved it, and you wouldn't have to recalculate the same problem. And that's a little bit like what your immune system is doing, right? So I set you this math problem, and I give you this period of time to work it out. You have to get your calculator out, pull your solution together, and then you answer me, right? And then I ask you the same question a bit later, and you don't need time to work it out again, because you just remember, I've already done this, right? The answer's a quarter. What are you doing, Joe, right? So that's what your immune system does in a way, right? It has this first infection, and it's spending this time figuring out how to fight the infection. And then it comes up with a good response with a specific antibody that will target that particular infection. And then if you re-expose it the second time to the same pathogen again, it's not going to rework out how to fight that pathogen. It's just going to reuse its solution from before and deploy that response harder and faster, as we saw before, and prevent you from getting sick, right? And clear the infection really quickly. Okay, so we've said that your immune system remembers past infections to stop you getting sick for a second time, and we said that it's the basis of vaccination. But memory is kind of a funny thing, right? And I'm thinking about my own memory when I say that, because we remember some things very, very well, perfectly even, though they occurred a very long time ago. So this is me on my first day of secondary school with my two brothers. I think one of them is on, so thank you for joining, Zach. But I'm the one on the left. You can tell I have dimples. The other two do not. But the point of putting this here is I remember this day really well, right? I remember how I felt. I remember feeling nervous. I remember meeting my teacher for the first time, the walk to school, meeting people who were going to become my friends. And it's sort of viscerally quite burned into my brain, right? This is not a day I'm going to forget. On the other hand, there are some things that we do very often that we don't remember all that well at all. And so here is a picture from my phone of the instructions as to how to open my mailbox at the university, right? You know, turn left a certain number of times and then right a certain number of times. It's actually a very difficult thing to do. But the point is I've done this dozens and dozens of times and I don't remember it, right? Every time I want to open my mailbox, I pull out my phone and I look at the picture because I don't remember how to do it without that prompt. So your immune system isn't very different than this, in the sense that it can remember some things very well and other things much less well. So the question really is COVID-19 like my mailbox code, or is it like my first day of school? Is it something that your immune system remembers really well, or is it something that your immune system remembers really poorly? Or is it somewhere in between, right? That's not a binary. There's a continuum of options between being remembered really well or not really well. And if we look at other pathogens, we can see that your immune system is exhibiting this continuum of memory. So a pathogen like measles, the measles virus is actually extremely well remembered by your immune system. So if you get infected with measles, your immune system will generate a response, and that response, and that specific response will be remembered for decades and decades. And it's why measles is a very good disease to vaccinate people against, right? We vaccinate them as children, and they have immunity lasting for decades and decades going forward from that point. And in fact, if you ask immunologists to estimate how long the immune memory of measles lasts, they might tell you up to 200 years, right? So this is an extremely good memory that your immune system generates of measles, and it's why you get vaccinated only at the beginning of your childhood, and you don't need to see the virus again to be immune from it. Tetanus is a bacteria that causes infections in people, and many of you may have had multiple tetanus shots over your life. In fact, it's quite common to have tetanus boosters every 10 years or so, because whilst your immune system is pretty good at remembering tetanus, it does need periodic reminders, and that's what those boosters serve as, to prevent you forgetting tetanus. So tetanus we could put somewhere in between measles and the last disease I'm going to talk about, which is just the common cold. Now, I should say that the common cold is actually quite a complicated collection of different diseases. It's actually caused by many different viruses, but each one of those viruses is actually pretty poorly remembered by your immune system. And that's not that surprising, right? Because we know from experience that we get colds year after year after year after year. It's not like being a senior citizen protects you against getting a cold. Unfortunately, people throughout their entire life get sick with colds. And that's because your immune system is not remembering this pathogen, so it can't reuse the response it used last time. It has to start from scratch every time. And as a result, you get sick, right? Because you have that delay period where your immune system is figuring out how to get rid of the disease. So where does COVID-19 fit on this continuum? Is it closer to the common cold end, the measles end, or somewhere in the middle? And that's really the problem that these authors in this paper try to shed some light on. Okay, so enough background, let's dive into actually what's done in this paper and what's really going on. So what do the authors do in this paper? I'm going to take a drink. Right, so what they're doing is they start off on the left here with some monkeys, and what they do is they initially challenge or expose those monkeys to SARS-CoV-2, the virus that causes COVID-19. And you can see the days being counted off on the bottom here from that period on the left, and I'm going to take you through the rest of it, but just you can see the days, and I've given you an indication that DPI stands for days post initial infection, and DPR stands for days post-reinfection. That second one will make more sense in a moment. So we've initially challenged or exposed the monkeys to the virus, and then we're going to monitor them to see if they get sick, which they should because we've exposed them to this virus, but also make sure that they get better, right? So we want them to experience the disease and then recover. Then we're going to take a subset of those monkeys and we're going to re-challenge them or re-expose them to the same virus, SARS-CoV-2, and then basically just ask, did the monkeys get sick, right? Because if they got sick, then it suggests that their immune system didn't remember the virus. And if they didn't get sick, then it suggests that the immune system did remember the virus. Okay, so that's kind of in broad brushstrokes what the authors have done here. But how do you do that? How do you go about monitoring these things? What's the measure of being sick or not sick? Well, there's a few different measures that the authors are going to use. The first one is swabs, and this is going to be familiar to you because people all over the world who are concerned that they have COVID are going into hospitals and getting swabs. And then what we do is we look for, scientists look for the presence of the virus on that swab. And that's exactly what the authors have done here with the monkeys. There's a periodic sampling of the monkeys where they're being swabbed to basically monitor to look for, can we find virus in the monkey after we initially infect it, after we try to reinfect it, and when it's sick and when it's recovered. So that's one thing we're going to do. Number two, we're going to look for antibodies. Now, I told you that antibodies are these sticky proteins that your body produces in response to a specific pathogen. So here we're going to be looking for specific antibodies. ABs is short for antibodies. Specific antibodies that respond to the SARS-CoV-2 virus. And we're going to look for them after we've challenged the monkeys initially and then after they've been re-challenged with this infection. So those are going to report on kind of what's going on in the monkey's blood with the antibodies and what's going on with the virus. But how do we know if the monkey's actually sick? Well, we can use the same things that we would use on people. We can use x-rays. So we can take pictures of the monkey's lungs to see whether they look healthy or whether they don't look healthy. Chest x-rays are going to be used by doctors. They are used by doctors all over the world to look inside your lungs to see, do you have pneumonia? Is there fluid in your lungs, which is going to be obstructing your ability to breathe and get oxygen from the air and breathe out carbon dioxide that your body is producing? So we're going to take them when the monkey should be healthy, when we think it might be sick, and then when we're interested to see whether it's sick or healthy because we wanted to see if its immune system remembered the virus. And then the last thing we can do is what's known as histopathology. And histopathology basically just means looking at the monkey's lung under the microscope, right? Having in detailed look at the microscopic cellular level, what's going on? Does the monkey's lung look healthy or does it not look healthy? Does it look like the monkey has got the disease or does it look like the monkey is in the clear and is pretty healthy? So those are the experiments I'm going to be taking you through, those 4 different areas, looking for swabs of the virus, looking at antibodies to see if the monkey's mounting an immune response to the virus, looking at x-rays to see how the monkey's chest looks, and looking at histopathology, looking at the monkey's lungs under the microscope to see whether they look healthy or not. Okay, so if that's what they did, what did they find? What did the authors show? So before I show you the data on swabs, I just want to explain to you how it's going to be displayed. They're going to be showed on these graphs. I'm sorry about the quality of the graphs. They're not wonderful. That's not my fault. It's the fault of the authors. Unfortunately, they, in the published work, the graphs are not fantastic quality. But along the bottom, we're looking at days post-infection, so the number of days that have elapsed since the exposure or re-exposure to the virus, in the case of DPR, which would be days post-reinfection. And then along the side, we're looking at viral loads in nasal swabs. And what that really just means is the amount of virus that we detected on the swab that we took from the monkey's nose. I should point out that along the side is actually a logarithmic scale. And you might be familiar with this with something like the Richter scale, which is used to measure how strong earthquakes are. Basically, what it means is as you go up the scale, the distances between the numbers increases. So the distance between 1 on this scale and 2 is actually only 1/10 as big as the distance between 2 and 3, and so on and so on. So really all that means is if you move up on a logarithmic scale, you're getting a lot bigger, a lot more quickly, right? It's just a way that we use of making, displaying data so that really, really big points don't block out really small ones. Cool. Okay, so that's taking you through the graphs. So let's look at the graphs. So I'm splitting the monkeys here into two groups, monkeys 0, 1, and 2, and I'm displaying the data about their swabs on the bottom left. And the other group is monkeys 3, 4, 5, and 6, which I'm displaying in the middle. And the monkeys 0, 1, and 2 were the monkeys we just infected once. So these monkeys are not going to be reinfected, whereas monkeys 3, 4, 5, and 6, we're going to attempt to reinfect them. We're going to see what happens. So in both cases, after we initially infect the monkeys, at day 0, you can see we don't detect virus in the nose of the monkeys. And then unsurprisingly, we see this big increase in the amount of virus that's being detected in the swabs from the monkey's nose. That's totally consistent with the monkeys getting sick. The virus is replicating inside the monkey's respiratory tract, and you can pick it up in swabs of the nose. And then also unsurprisingly, the virus is reduced, right? Around day 10 post-infection, you can see that the monkey has successfully fought the virus off, it looks like, right? Because we're not detecting virus in the monkey's nose anymore. It looks like all the virus has been killed by the monkey. So what's going to happen when we attempt to reinfect the monkey? Well, let's have a look. So at 28 days post-reinfection, if you look at the middle here, that dotted line represents the point at which we re-expose the monkeys to the virus. And what you can see, quite remarkably, is that there's absolutely no increase in the amount of virus detected in these monkeys, monkeys 3, 4, 5, and 6, that is after the re-exposure. And that's in stark contrast to what happened the first time we exposed the monkeys to the virus, where you saw this big spike in the amount of virus that was detected. Now, on the right here, what I'm showing you is the comparisons of the reinfection amount of virus detected and the initial infection amount of virus detected at specific days. So you'll see the first two bars refer to day 0, the second two to day 5, day 7, day 14, post either infection or reinfection. And the blue dot represents the bar which is in the initial infection situation, and the orange dot represents the bar in the reinfection situation. Now, the reason in the first two you can't see any bars is because they didn't detect any virus at this point, right? And then at day 5, you can see under the blue dot, there is virus detected in the initial infection situation, but in the reinfection situation, there's no virus detected. Same is true at day 7, and at day 14, there's no difference because under both situations, there's no virus detected at this point. The monkeys who were initially infected by day 14 have successfully cleared the virus out of their system, and so you can't find it anymore. So interestingly, this is also replicated. I say interestingly, it's pretty unsurprising actually. It's also replicated if you swab the monkey in other places. If you take a swab from the monkey's throat or a swab from the monkey's anus, both of which are places that we'd expect to find virus during an active infection. And you can see that what's happening here is the monkey initially will see a spike in virus detected by these swabs, but when we reinfect them in the central two panels, you can see that there's no virus detected after we reinfect the monkey, or we attempt to reinfect the monkey. So collectively, I think this gives you a picture that after we initially infect the monkey, it looks like there's virus being produced, and we can pick it up in swabs of the monkey's nose and the monkey's throat. But if we allow the monkey to recover and then re-expose it 28 days after the initial exposure, we can't see virus replicating, which suggests that the monkey is successfully fighting the virus off and preventing it from replicating and establishing an infection. Cool. So we talked about the first one of my 4 measures. Now let's talk about the second one, which is looking at antibodies. Okay, so we're looking at antibodies at various different points along the progression of this experiment. And what you can see here is, again, I'm talking about monkeys 0, 1, and 2. These are the monkeys that were infected only once at the beginning at day 0. And you can see that, excuse me, as the experiment moves on, there is this little lag, right, where the monkeys are trying to generate the antibodies. That's the gap in the plots I was showing you earlier. The monkey's immune system is looking for the right antibody to fight this virus off, looking, searching. It finds it, and then you can see it's produced. beginning around day 14 and then into day 21 and 28. And you can see the relatively high levels of the antibody present. So what about in the situation where we reinfect the monkey? Well, what you can see is the first half of this graph is basically the same, right? We've initially infected the monkey. You can see it's looking for the antibody, looking. It finds it around day 14. We get production of the antibody. And then around day 28, when we reinfect these monkeys, you can see that they then kick into a higher gear, right? They're producing even more antibody. And there's this enormous peak at day 42 post-initial infection, which is 14 days after the second infection. And the antibody levels at that point are much higher than they were 14 days after the first infection. And that's what's plotted on the far right. You can see 14 days post-initial infection, you've got some antibody produced. But 14 days post-reinfection, you have even more antibody produced, which suggests, I mean, that backs up what I was telling you earlier about the idea of the immune response kicking in quicker and more punchy, and it's being more effective and more strong the second time around. And that's stopping the monkey getting sick. And so these monkeys don't look like, in terms of the antibodies, they look like they've mounted a really effective second response the second time we've exposed them. And it looks like that is the basis maybe of why they're not getting sick. Right, but so far we haven't actually taken a look at what's happening in the monkey's lungs. So let's now do that. So let's look at some X-ray pictures. We're going to look at first an X-ray picture from day 0. This is taken before we expose the monkeys to the virus in the first place. So this should be a healthy monkey lung. And you can see, hopefully down the middle, you can see the spine of the monkey, you can see the ribs coming in from either side, and you can see the monkey's heart and major blood vessels in the middle. Now, X-rays, first of all, I should say I'm not a radiologist, and people train for a very long period of time to look at these X-rays and be able to interpret them properly. But what I do know is X-rays of the lungs should show up in dark if there's lots of air, right? Because it means that the X-rays move through the lungs more easily, and so that shows up as a black color on the X-ray. And so it's good to have lots of darker color on your chest X-ray because it means that your lungs are full of air, and that's what they should be full of. Unfortunately, it looks like after 7 days post-infection, if you strain your eyes and look very closely, or if you're a radiologist, I'm told, you can see there's beginning to look like some cloudiness, and that's particularly pronounced in the red circle that the authors have kindly drawn on this X-ray for us. Now, physicians refer to these as ground glass opacities. And really what they are is it's the production of fluid and inflammation in the lung, which is basically replacing the air in the lung and causing the chest X-ray to look lighter rather than darker because it's not moving through air. Okay, so that's what's happening when the monkey's sick. That suggests that the monkey's developing pneumonia when it's sick. Again, that's not surprising. That's what happens with a respiratory infection like SARS-CoV-2. So let's look at what happens when the monkey gets better. At day 28, immediately before we re-challenge it with a new specimen of the virus. So what you can see here is in the blue shaded circle, the lung looks clearer here, doesn't it? So we can see less cloudiness. It looks like there's not fluid appearing in the lungs, suggesting the monkey has got rid of the pneumonia it maybe had, and it's kind of on the way to being healthy again. And then if we look after we reinfect it, 5 days after we reinfect the monkey, that clearness remains on the chest X-ray, suggesting the monkey did not develop pneumonia the second time around. So the picture we're getting from X-rays is we initially infected the monkey, its lungs got filled up with fluid and it got pneumonia, it recovered, and when we reinfected the monkey, it doesn't look like that pneumonia developed. It looks like the monkey's lung remained relatively healthy and clear, and that suggests it didn't get disease. It wasn't really sick the second time around. So finally, let's have a look at the monkey's lung itself under the microscope. So what I'm showing you here are two pictures from the monkey's lung. On top is a sort of zoomed out version of the same picture on the bottom. And I've annotated to show you two important things in this. The first is that there's a lot of white space on this picture, and that's because there's a lot of air in the lungs, right? That's how it should be. The lungs are full of air. They're the organ that does allows oxygen into the bloodstream and excretes CO2 out of the body. And then we have these thin walls around the edge of the air sacs, And you'll have to take my word for it that those are full of blood vessels, and they're very thin, and that's important because it means that the oxygen in the air sacs can move very easily into the bloodstream, and the carbon dioxide in the bloodstream can move very easily into the air sacs to be excreted and breathed out of the body. Cool. So what happens when we look at some monkey lungs taken from 7 days after the initial infection? What do they look like? Well, it's not a pretty picture. We've lost a lot of the clear white space that should appear on the micrograph, and that's indicative of the air sacs getting much, much smaller because there's this massive inflammatory response in the lung. We've got much, much thicker walls of the air sacs, smaller air sacs, and it doesn't look like a pretty picture. It looks like this monkey is going to be having trouble breathing. It's certainly not in a healthy place. And if we compare that to a similar lung looked at, but we're now looking after we've reinfected the monkey, after it's recovered and we've reinfected it, what does it look like? Well, it's a much, much better looking lung. We've still got nice big air sacs. Maybe it looks a little bit different. It looks a little bit more sort of inflamed, engorged than the control, but it's certainly a hell of a lot healthier than the lung in the middle. So if we combine this with the X-ray, the sort of picture we're getting is healthy lungs at the beginning. After initially infecting the monkey's lungs unfortunately become diseased. We get inflammation, the air sacs get squashed out, and so the lung isn't working so well. We allow the monkey to recover, we reinfect it, and the lungs don't look bad when they're reinfected. They look basically normal and basically quite healthy. So collectively then, I think the authors have pretty convincingly shown that monkeys do not get reinfected by SARS-CoV-2 after a second exposure 4 weeks after the first infection. And we know that because when we re-expose the monkeys, we don't detect virus in swabs from the monkeys' noses and throats. We don't detect pneumonia in the X-rays taken of the monkey's lungs, and the monkey's lungs look healthy under the microscope. Cool. So that's what the authors have shown. How does that fit in with what we already know, and what questions remain outstanding? Well, I think the most important question that remains outstanding is to do with timing. And this is a really difficult question to answer. It's not that scientists haven't tried to answer this question or don't have good experiments for addressing it. It's just it's very difficult to do these experiments. Ideally, we'd like to know if we infected a monkey 3 or 4 years ago, or 5 or 10 or 15 or 20 years ago, and we re-expose it to the same virus, does it get sick? We'd like to do this experiment with a time gap, not of 4 weeks, but of 4 years or 4 decades to understand how long the immunity is lasting. How long is that memory lasting? But of course, that's not possible with a virus like COVID-19, which has emerged in the past 6 months. We didn't know that this virus existed 5 years ago. We couldn't have been infecting monkeys to do these experiments 5 years ago because we didn't know that the virus existed. So we're severely limited in our ability to look backwards to enable us to do these experiments to address how long immunity really lasts. But we do have an important data point from this experiment, which suggests that at the very least, your immune system is capable, or the monkey's immune system, which is extremely similar to yours, is capable of remembering an infection full stop. And then there's a second question to do with whether immunity is going to last in its entirety or whether it might be remembered in some degree. So even if immunity expires fully, are you protected by some degree by the memory that you have? So is it, even if you get sick a second time, is it possible that you get sick less severely than you got sick the first time you got infected? Because these memories are not a binary, yes, you remembered, no, you didn't remember thing. There's a continuum. And then, of course, we need to think about the public health considerations of this. If we do develop a vaccine, are people going to need regular treatment with this vaccine, or are they going to be able to be vaccinated just once? And can we use a measure like antibody levels to predict whether someone needs to be vaccinated or not? Cool. So if you weren't listening to anything I just said over the last 40 minutes, what's the answer to the question, can I get COVID-19 twice? Well, what I would say is this: in the short term, it's extremely unlikely. We've seen that from the monkey. Over a period of months, it's unlikely that your immune system is going to forget it. The monkey's immune system certainly did not. But we're not sure about when immunity might expire in the medium term or the long term. because we haven't been able to do those experiments yet, we didn't know this virus existed more than 6 months ago. And if we look to the best evidence that we can kind of speculate with, it would come from looking at viruses which are similar to SARS-CoV-2, like the original SARS virus and MERS, Middle East Respiratory Syndrome. If we look at those viruses, we have some data. It's not foolproof and it's not comprehensive, but we have some data suggesting that immunity lasts around 2 to 4 years for these related coronaviruses. So it wouldn't surprise me if it was a similar kind of time length with this virus, but we don't know. It is different. It's a different virus, and it could be longer, it could be shorter. That remains for scientists to establish. Rest assured, there's lots of people pretty hard at work on this problem. So that's it from me. I'd just like to say a big thank you to the whole of the D4P team from all over the world and the US, and invite questions from any of you guys if my talk has spawned them. So thank you very much.